Introduction to enzymes. This presentation is related to the basic definition of enzymes and the classes of enzymes. Enzymes basically are important components present in all living cells. It comes from a Greek word enzymos, meaning leavened. It also means in living, that is in and zyme, living cells. So these are actual agents which have been discovered to spark reactions, that is to initiate and speed up different kinds of reactions. Now looking at enzymes, their nature are globular proteins. They are catalysts of biological systems. That means they enhance the reactions, the rate of a reaction. And most of their names ends with ASE. So examples, we have lactase, amylase, dehydrogenase and oxidase. So when we speak about metabolic reactions, there are two types of reactions. Anabolic reactions, which mean the building up reactions, and catabolic reactions, meaning that they are the breakdown reactions. So when I say anabolic reactions or build-up reactions, this is where small molecules build up together using energy, as you can see here, to form larger molecules. So this is called as an endergonic reaction because energy is required. On the other hand, we have catabolic reactions, which are breakdown reactions, as I mentioned, of larger molecules breaking down into smaller molecules. So here energy is expended or it is released. So these reactions are called exergonic reactions. However, both the types of metabolic reactions require enzymes. That is the point that has to be noted. So we require enzymes for all kinds of metabolic reactions, whether they are anabolic reactions, that is build-up reactions, or catabolic reactions, that is breakdown reactions. Now coming to the classification of enzymes. It is not that the enzymes are functioning just like that. There are different specific classes performing specific reactions. So the classes go like this: oxidoreductases, transferases, hydrolases, lyases, isomerases and ligases. We will look at each one of them, but this slide is one which shows a one-line definition of the different major classes of enzymes. So as you can see, oxidoreductases will catalyze oxidation reduction reactions, which are also called as redox reactions. Transferases are a class of enzymes which can transfer functional groups between donors and acceptors. The class of enzymes called hydrolases will catalyze the hydrolytic reactions or hydrolysis of the substrates. Lyases are a class of enzymes which catalyze removal of a group of substances other than hydrolysis. Isomerases catalyze intermolecular rearrangement of substrates. And finally, we have ligases which catalyze the union of or forming together, bridging two molecules. Now, one by one, let us look at each one of these enzymes along with their examples. So here we see that oxidoreductases are enzymes that catalyze oxidation reduction reactions, as I mentioned earlier, between two substrates. So if the two substrates are A and B, we see that there is a transfer of hydrogen, that is oxidation and reduction reactions between one substrate and the other. Example, oxidases, dehydrogenases and reductases. The next class is transferases, where you see there is a catalyzing the group transfer reactions. These are of the general form A plus B, where we have one group called X. So this X could be transferred to B in these reactions, catalyzed by enzymes called as transferases. Example of transferases are transaminases. These transaminases are enzymes which will transfer amino group. So if this X is an amino group, that is a NH2 group, this would be transferred to the next substrate using the enzyme transaminase. Similarly, we have kinases, another transferase group of enzymes where the phosphate group, if X is the phosphate group, it will be transferred to the next substrate. So these are transferases. Next come the group called hydrolysis. As I told you, these will catalyze hydrolytic reactions. That means water plays an important role in these kinds of reactions. So we have a substrate AX which needs to be hydrolyzed, or whenever there is a need for hydrolysis reactions, water will split itself as OH and H, as you can see, and the entire molecule is now hydrolyzed and no longer remains as AX. So these hydrolyses we have specific examples like lipases, esterases, amylases and proteases. The group of enzymes which are called lyases will catalyze removal of a functional group. So here we have functional groups like X, it could be one functional group, Y could be another functional group. So these are removed away, and in the process what happens is there is a linkage between A and B, as you can see with the double bond. That is what I have mentioned as this is a non-hydrolytic reaction. You do not see the role of water in these kinds of reactions, isn't it? So we have created, however, a double bond between the two substrates. So here we have decarboxylases, aldolases, dehydrases, deaminases and synthases. All these belong to the major group called as a lyase. Okay, and the next one is an isomerase. These catalyzes isomerization reactions, including epimerization and cis-trans isomerizations. Ligases catalyzes the synthesis of single bonds. Earlier we saw double bonds coupled with the breakdown of energy. So when we have two different substrate molecules, bringing them together into one whole large molecule is what is the role of or function of enzymes called as ligases. In this process, energy is required because a bond is formed in order to form a larger molecule. So this energy is usually utilized by the breakdown of ATP to ADP and inorganic phosphate. When I say utilization of energy, A plus B utilize ATP, which is the chemical form of energy, and in the formation of AB, this ATP gets broken down to form ADP and inorganic phosphate. Examples are synthetases and carboxylases.